Our next presenting company is going to be Juventas, so I'm looking for Dr. Aras. Oh, okay. I think we have the next best thing, Dr. Penn. So, one really good question when we talk about cardiology is, why are we taking cells from your marrow, taking them out, and putting them back in? Well, one answer might be because the cells are not tracking there naturally. What is the mechanism by which they track? This is a very, very interesting company that's really looking at the SDF1 mechanism, and I think you really want to pay attention to it, and the fact that they're in phase 2 or 3 phase 2 clinical trials. It's something, worth paying attention to, and while it's a private company today, that may not always be the case. Dr. Mark Penn, who you should recognize from the cardiology panel this morning. Thank you.
Thanks, Jason. And Rahul, the CEO, is not here, but I'm the founder and CMO, so hopefully I know something about what we do. Just as an executive summary, we were created in 2007 with an exclusive license of my technology when I was at the Cleveland Clinic and more from Summa. Clinical stage biotech company, as Jason said, 3 phase 2 trials ongoing. Our lead product is JVS-100, which encodes a human form of SDF1, otherwise known as CXCL12, and it's, again, the subject of trials not only in heart failure but also in critical ischemia, and we have a pipeline that provides opportunities to develop standalone therapies and adjunctive therapies for cell-based strategies as well as standalone SDF1 therapy. The hypothesis that has driven my laboratory, and hence Juventas, the laboratory since July of 2000, is the concept that I mentioned earlier today. There is an endogenous stem cell system that does try to repair our body when we're injured. And we believe that the reason it's not clinically beneficial to us isn't because we lack stem cells. It's the fact that the signals are dysregulated in a way that we don't optimally heal. Now, said a little differently, and a corollary to that hypothesis, is the concept that we as mammals have evolved to scar instead of heal. And we believe that's in an attempt to minimize tumor. But it's that kind of understanding and hypotheses that has driven Juventas. And we believe that by reintroducing those factors that are dysregulated, we in fact can induce that endogenous stem cell repair pathway and lead to clinical benefit. And we would suggest that a lot of forms of adult cell therapy, in fact, is releasing SDF1 into the tissue and inducing that endogenous system. So fundamentally, we believe that adult cell therapy works based on this simple plot. We know that SDF1, the key molecule for recruiting stem cells to the site of any injured tissue in the body, and that's our work in many other groups, is up and down in about a week. It's a very short-lived signal. But interestingly, in the end organ that's injured, be it the brain, the heart, the kidney, or the liver, or frankly the skin, CXCR4, its receptor, does not come up until about 3 to 5 days post the injury. So in fact, we have a signal that recruits cells, and the organ that's been injured doesn't respond for about till that signal is almost waning. True also for the bone marrow, and one of the reasons why CD34 cells work so well is that they're delivered near a time when there's a lot of SDF1 expression, so they in fact can respond. What we believe adult cell therapy does to that point is it temporally realigns SDF1 with its receptor CXCR4, because we either reestablish sometime late out here, because in the heart, the CXCR4 stays up forever. Or at the time of acute MI, we extend SDF1 to a point where CXCR4 is now upregulated, and we lead to a lot of improvement in tissue outcome. And that's an overall hypothesis that certainly in mice we've proven without a doubt, and we believe it's active in humans. Our lead product, JVS-100, is a cDNA plasmid. It's naked DNA that encodes SDF1. It yields a therapeutically relevant expression of SDF1 for about 17 days. And what we know in the lab is to get a benefit in heart failure, you need to induce that expression for about 12 to 14 days. It's direct delivery to the damaged tissue. It's highly scalable because it's plasmid. It's demonstrated safety profile, and it's stable for at least 24 months, and I think we now have 36-month data at 20 degrees. And as I probably shouldn't tell, but I tell my teenage kids, if you can make beer, you can make this plasmid. So what we believe we've really done is we've taken cell therapy, bone marrow harvest, isolation, maybe propagation, and we've turned it into a vial. A drug that you can deliver to the tissue that will induce the body's own stem cell response and try to induce true tissue repair, if not replacement, in a time period that's safe, highly scalable, cost of goods substantially less than any cell therapy product. And again, can be in the cath lab, can be in the operating room, can be anywhere it needs to be. Overexpression of SDF1 induces the endogenous stem cell system that we clearly have demonstrated through proliferation of the end organ stem cells, so in the heart, cardiac stem cells, in the brain, neuroprogenitor cells, and it induces the recruitment of bone marrow cells. We believe our data suggests that the bone marrow cells induce new vasculogenesis or vessel growth, and the end organ specific cell can induce new neurons, potentially new cardiac myocytes, new hepatocytes, and things of that nature. But you can also enhance exogenous cell therapy by delivering SDF1 along with those cells, because that SDF1 signal, we've shown in preclinical studies, leads to greater survival of your cell product and leads to greater recruitment of then the endogenous cells to aid and help that cell product. So where is Juventas now? We're in phase 2 for heart failure in 2 phase 2 programs, one through direct injection using the biocardiohelical catheter, the other using retrograde delivery. It's the first in the U.S. retrograde study for drug delivery. We have a critical limb ischemia trial. We have preclinical data that would support an IND for acute MI and chronic chest pain today, and we have some early studies in sphincter dysfunction looking at muscle regeneration. JVS 200 is monocyte chemotrectic protein 3, or MCP3, which is a mesenchymal stem cell homing factor that would go along with the MSC product of somebody else's, and we have several discovery projects ongoing. Looking at our clinical program, heart failure, I don't have to tell anyone here, is a huge market. As a physician, it's a huge unmet clinical need. There's at least 1 million symptomatic heart failure patients in the United States today. You can double to 2.5 times that for worldwide. About $37 billion spent annually on heart failure in this country. And again, the target population that our clinical trials are designed to focus on is truly the symptomatic heart failure patient who has significant inabilities to maintain their average daily living activities. Our phase 1 heart failure trial was a 17-patient open-label dose escalation study. The primary inclusion criteria was history of a myocardial infarction with an EF under 40%, New York Heart Association Class 2, with wall thickness large enough to take the needle more than 0.5 centimeter. We had one treatment with 5, which we thought would be sub-efficacious, 15 or 30 milligrams via the endoventricular injection catheter. We met our safety endpoint. There were no adverse events related to the drug or the delivery, and 15 of the 17 patients survived at least 18 months. 2 patients died about 30 to 35 days post-treatment. One, an elderly woman who just succumbed to heart failure after she stopped all her meds, and the other gentleman who had true sudden death. Efficacy-wise, we had, although we would have anticipated an additional 4 deaths in that 18-month period. Efficacy, dose-dependent improvements in quality of life, 6-minute walk, and NT-proBNP, or the biomarker for heart failure. We had dose-dependent stabilization of ejection fraction and left ventricular end systolic volumes. So while these parameters did not improve, they did not decrease as they did in the low dose. And we had at least 50% of the patients in the 15 and 30 milligram doses improved at least one NYHA class, and these data were all published in Circulation Research earlier last year. Showing the 12-month data, what we see is, in fact, the 5 milligram sub-efficacious dose patients stabilized or got worse as far as their BNP. In the 15 milligram dose, some improvements, particularly in the quality of life. And in the 30 milligram dose, we see at 12 months improvement in BNP, improvement in quality of life, and a trend towards improvement in 6-minute walk. So a nice dose-dependent response, which we believe shows the indication that SDF1 is in fact mechanistically linked to left ventricular remodeling. We then launched the STOP-HF trial. So 93 patients. Last patient was enrolled in October of last year. Placebo-controlled, randomized, blinded clinical trial looking at inclusion criteria. It's very similar to the phase 1. Primary efficacy point is 6-minute walk and quality of life. It's done at 16 sites throughout the U.S., and these data are targeted either later the next quarter and the 12-month data out later this year. Again, we asked 2 questions. Is is treated better than control, and then at 12 months is 30 milligrams better than 15, as we saw in the phase 1. The other delivery strategy we're looking at is retrograde. One limitation of endocardial injection is the fact that you need to have wall thickness thick enough to take a needle safely. About 20 to 25% of significant heart failure patients have walls that are too thin, and we're unable to treat with the biocardial catheter. Therefore, retrograde via the coronary sinus, all on the right side of the heart, is a safe right-sided procedure or less stroke risk, and we worked very hard to get IND approval for a retrograde study in the United States, again, the first for drug delivery. The balloon's inflated to occlude the coronary sinus. We infuse JVS 100. There are no wall thickness issues. It's 15 to 20-minute procedure. Broader physician, utility-based heart failure docs, electrophysiologists, general cardiologists, as well as interventionalists can do it. It's not purely an interventional procedure. And there's several of these balloons that are on the shelf at most hospitals. So, in fact, cost of goods wise and things of that nature and access to the catheter is very simple. We designed the retro heart failure study. It was a phase 1, phase 2 study. The reason for this design is, again, the FDA had not seen a retrograde study in the U.S. They wanted a phase 1 open label study. That is now complete, and we're well into the 60-patient phase 2 study. Again, looking at a little bit higher dose, 30 milligrams to match the STOP-HF, which we believe would be the most efficacious in that study, and then extending our dose range up to 45 milligrams. Preclinical studies suggest that, interestingly, retrograde and endocardial injection, in fact, give very similar results at very similar doses. We also have a critical limb ischemia study. In our phase 1, I failed to mention, we did stress testing at baseline at 4 months post-treatment. The stress difference score, the amount of ischemia in the myocardium, was 4. That, to a cardiologist, is a lot of ischemia around old infarct, but not need for bypass or stenting. In the 15 and 30 milligram doses, that stress difference score of 4 went to 0. We completely revascularized the border zones, suggesting we can significantly increase the microvasculature of tissue. That led us to critical limb ischemia, where there's a huge microvascular problem on top of the macrovascular problem. Peripheral vascular disease affects 18 million people, estimated 3 million diagnosed in the U.S. with 200,000 cases annually. About a third undergo amputation in the first year. So leading cause of amputation outside military-related trauma, and it's a very expensive treatment for the country. The STOP-CLI trial was a phase 2A trial, 48 patients, 4 cohorts looking at 8 milligrams in 2 ways, 4 milligrams in 16. This trial has been completely enrolled in the United States and India with the full 1-year follow-up data set due out in Q3 of this year. We've had good evidence in the early data out of good SF36 data, including the VAS score and things of that nature. So we're pretty excited to see the full data set. So we've treated over 125 patients with over 750 months of follow-up and cumulative safety data that all look very good. The STOP-CLI testing in patients with critical limb ischemia, again in Q3, STOP-HF and with JVS 100 and heart failure in Q2, and retro-HF due out in Q3 to Q4. So we have a very exciting data-driven year that we're excited to see the results. Just to show you a little bit about enhancing the cell therapy side, again, we can enhance and we can take out stem cells, we can propagate them, we can transfect them or co-deliver JVS 100 to aid in the engraftment and the survival of those cells. We have a system where we can actually induce SDF1 in cells with a peak around 10 days, but get that 17-day expression. When we looked at modified MSCs, we about tripled the amount of SDF1 they make. MSCs make it already. And we can, in fact, greatly enhance the recruitment of cardiac stem cells. We showed a number of years ago that MSCs and SDF1 recruit CSCs more than just MSCs alone. Furthermore, we can double the survival of these cells. We'd still lose 50% over time, but in fact, here you see we double the amount of engraftment and survival of the cells at the beginning, so over time we have greater number of MSCs surviving. And if we look at the functional effect in an acute rat MI model, here's pre-injury, here's no treatment, they drop their EF pretty significantly. Here's MSCs, significant benefit but modest. MSCs that overexpress SDF1, greater vascularization, greater survival of those MSCs, greater myocyte survival. We can, in fact, significantly enhance that mesenchymal stem cell therapy. So in summary, JVS-100 encodes SDF1, which has been shown to improve organ function in multiple different areas of organ injury by recruiting stem cells, causing neovascularization, organ-specific stem cells, remodeling of scar, so multiple different mechanisms. It's safe and preliminary efficacy has been shown. We have ongoing clinical trials with more than 100 patients dosed with no known side effects. Cells engineered to overexpress SDF1 is really a very interesting way to get next-generation cell products, lower cost of goods, enhance efficacy. And Juventas team collaborator partnered on FDA allowance for the first retrograde study, as well as the first modified cell therapy study that ultimately was approved by the FDA, but never executed by our partner, and we now retain all the rights to that that approach. So homing factors have efficacy, they have cell retention and engraftment strategies, and a broad therapeutic window, and we're pretty excited about what inducing this repair system can do for the body. And in summary, studying the endogenous stem cell repair system, I think there's a lot of great work done with stem cells, but understanding that biology that regulates stem cells, I believe really can identify novel druggable targets that will allow us to induce a lot of tissue repair independent of having to give the cell product. And, you know, my group in particular has really focused on what's the biology behind these things, not just the cells themselves, and I think it's a very exciting future for us. Thank you very much.